Hello, my name is Pablo Camara. I'm from the University of Pennsylvania, and the title of this talk is Single-Cell Transcriptomic Analysis of Highly Multiplex Cytometry Data via Antigen Mapping. This talk is about immunophenotyping, namely the study of heterogeneous cell populations by profiling their protein expression at the single-cell level with antibodies. And this has been key to our understanding of tissue cell composition and function over the past years. It has found many applications in multiple areas of research, in particular in the context of the study of autoimmune diseases, cancer, and immunology. These applications have led to the rapid development of quite amazing technologies that can phenotype thousands of cells based on dozens of proteins. Traditionally, those technologies have been non-spatially resolved. Two classical examples would be flow cytometry and mass cytometry, or CyTOF. Flow cytometry in standard applications can be used to profile up to 20 simultaneous proteins, whereas mass cytometry in standard applications can profile up to 40 simultaneous proteins. However, by using some of the most recent implementations of these technologies and optimized approaches, one can in principle go well beyond these limits. In the past decade, several other technologies of this type have emerged that in addition can perform spatially resolved measurements. So these technologies can profile up to around 30 simultaneous proteins in a tissue section in a spatially resolved way and with single cell resolution. There are few examples of those technologies. Some of the most popular ones are co-detection by indexing, or CODEX, imaging mass cytometry, which has been there already for some time, and digital spatial profiling. Currently, the standard way of analyzing highly multiplex cytometry data produced by these technologies is using automated gating approaches. There are many algorithms to that end. For instance, EXIF, Phenograph, SPADE, BISNI are some popular ones. And all these methods, they perform dimensional reduction of the protein expression space followed by clustering. However, the user still needs to manually annotate the resulting clusters with biological terms based on the presence or absence of protein combinations. So here we have, for instance, some example of a real data set that has been analyzed with the SPADE. And in this representation, each node corresponds to a set of cells that has been clustered together based on their protein expression profile. So basically, the user would need to go through the entire representation looking at what combinations of proteins are expressed in each node, and based on those, it would annotate with biological terms, such as name of a cell population, each of the nodes in the representation. So obviously, this is a very labor-intensive process that has some limitations. First of all, it requires well-curated antibody panels that only include markers for which we understand very well what populations in the tissue are going to express that marker. Moreover, it's hard to annotate nuanced cell types and states, in particular if there is no specific marker in the panel. So consequently, very often we will end up with situations where some of the cell populations that were originally present in the tissue are not captured in the analysis, and also with cases as in here where we have multiple clusters that have been annotated with the same label. Those clusters differ little on the profile of expression of the proteins that we are measuring. However, it's very hard to manually interpret those differences in biological terms. So the question that we would like to address is the following. Can we leverage single-cell RNA sequencing datasets to automate this process of identifying, annotating, and gating cell populations in highly multiplex cytometry data? In single-cell RNA sequencing, the expression level of thousands of genes is profiled for each cell, and this provides a very high resolution in cell type and state identification and definition. Moreover, hundreds of new data sets are being produced each year for the past few years, and there are large ongoing efforts, such as the Human Cell Atlas, to comprehensively characterize the cell composition of tissues under different conditions. In addition, some of the most recent approaches to single-cell RNA sequencing, such as CytSeq, can be used to not only profile the transcriptome of individual cells, but also concurrently measure the protein expression level of a handful of surface proteins. And this is done by staining the cell suspension with antibodies that has been linked to polyadenylated and barcoded oligos, and which in this sense they mimic endogenous mRNAs. So this method can be therefore used to determine the relation between gene and antigen expression. So basically the question is, can we leverage all these resources to facilitate and automate the identification, annotation, and gating of cell populations that I was describing in the previous slides for highly multiplex cytometry data? To that end, we have developed a computational method that we call Stevia for spatial transcriptomics via epitope anchoring. Stevia takes as input a highly multiplexed cytometry data set that we would like to annotate in an automated way, and a reference site-seq atlas from the same tissue. And Stevia will transfer the gene expression profiles and the annotations, the cell types from the site-seq atlas into the cytometry data set. To that end, it's important that the antibody panels that have been used in the site-seq atlas and in the cytometry dataset that we would like to annotate share a sufficiently large number of antibodies. Stevia comprises several steps. The first step is to apply a mixture model normalization scheme to the protein levels of each of the two datasets in an independent way, to eliminate or to reduce the background that is specific to each of these data sets. And this is important because although we are measuring basically the levels of the same proteins with in the cytometry data set and the site-seq data set, we are using two very different technologies to that end. On the site-seq side, we are basically sequencing the oligos that are attached to the antibodies. On the cytometry data set, it will depend on the specific technology and on the specific data set, but typically will be fluorescent levels. So this leads to very different backgrounds that we would like to remove using this mixture model, so that it will be easier then later to integrate the signal components. Here we have a specific example. This is CD4 protein expression levels measured with codex in a tissue section of the mouse spleen. These are the raw levels on the left. Here we have the distribution of the protein levels, and where we have fit this Gaussian mixture model, where one of the components corresponds to the background and the other to the signal. And on the right we have the same tissue section, but now labeled just based on the expression levels that corresponds to the signal component. So we see that this approach really highlights the signal, and in this specific case it highlights the T cell zones expressing CD4. Once it has normalized the protein expression levels in this way, the next step Stevia does is basically consolidating the protein expression spaces from site-seq and the cytometry data set into a single consolidated protein expression space. To do this, we are using the same general principles of mutually nearest neighbors and canonical correlation analysis introduced by the groups of John Marioni and Rahul Satya. And once we have a consolidated space, then we can basically transfer cell type annotations and gene expression information from the site-seq dataset into the cytometry dataset. So to that end, basically what we do is to look for each for each codex cell, what are the closest site-seq cells in this consolidated space, so namely which cells have, after all these corrections, the closest antigenic profile, and then we assume basically that those set of cells can be identified based on their antigenic profile, and in this way we can now infer under these assumptions what is the expression profile and the cell type that is assigned to those codex cells. The last step is to cluster the mRNA CytSeq data, but we would like to do it in a way that we take into account the mapping uncertainties so that the resulting clusters in the mRNA space are such that they differ enough in their protein expression profiles so that they can be mapped into the cytometry dataset in a confident way. If we were just clustering the mRNA space in a standard way, we would maybe end up with clusters that sometimes they do not differ enough in their protein expression levels for the proteins that we are profiling with the antibody panel, so that we cannot really distinguish them after mapping them into the cytometry dataset, and vice versa. Sometimes we will have clusters in the mRNA data that maybe we could have split further into smaller clusters, and still we would be able to distinguish those in the cytometry dataset after mapping it. So what we have done here is basically to develop a new clustering algorithm that is based on hierarchical density-based clustering, but where now the cost function is not given in terms of the mRNA, but rather in terms of the modularity of the mapped clusters in the cytometry dataset. To benchmark stevia and to illustrate the type of analysis that it enables, we have developed a CytSeq atlas of the mouse spleen, comprising a panel of 30 antibodies. And here what we are seeing is just the mRNA part of this CytSeq atlas, where we have performed just annotations based on clustering and differential expression analysis, and then we have refined those annotations by identifying gradients of expression that are significant as compared to some random model using some method that we recently published in PLOS Bio. So we have used this CytSeq atlas to annotate in an automated way using stevia several published highly multiplexed datasets of the mouse spleen. Here we have an example. This is a codex dataset of the mouse spleen published by Goltsev et al. In 2018. And here we see the UMAP representation of the protein expression space of this dataset, where the individual cells have been annotated according to populations from the CytSeq atlas identified by stevia. So now we have basically for each cell, we have its cell type assignment, we have its location in the tissue section, and we have the gene expression profile that has been assigned to that cell. With that information, now we can do many analyses. We can, for instance, look for colocalization patterns between cell populations to try to infer interactions between those populations. We can infer spatial patterns of gene expression for any gene that is contained in the CytSeq atlas. Here we have two examples of two genes for which we have predicted the spatial distribution of their expression using stevia, and on this side we have the experimental validation of those predictions using RNA-fish. And we can look also at the spatial colocalization of the expression of genes that code for paired ligands and receptors expressed in different populations, and we can do that in order to infer the way that different cell populations are interacting with each other. So here we have one example of some of the interactions that we have predicted using stevia on that codex dataset by leveraging that reference CytSeq atlas that we have generated. This is another example. This is a different dataset. It's a CyTOF dataset of the mouse spleen, also from the same paper. And on the left, we have UMAP representation of this data set where we have performed a standard analysis. We have run xIF to cluster basically these cells based on their protein expression profile, and then manually we have been annotating each of these clusters with the same annotations that in the original paper. And on the right, we have the same data set, but now annotated automatically by Stevia by using the reference CytSeq atlas that we have generated, and you see that the annotations are consistent, but also richer than the what would be a standard manual analysis. So the way we envision the future for computational analysis of highly multiplexed cytometry data is, and some of the lines that we are currently working on, is that there will be some efforts to integrate and harmonize different single-cell RNA sequencing data sets that are being produced and CytSeq data sets to build a set of reference atlases that will be stored in an online database. So then when a researcher wants to perform a highly multiplex cytometry experiment, he will be able to use a software like Stevia to connect into this database, select for a reference atlas or set of reference atlases that are appropriate to its experiment, and then Stevia will use those atlases to transfer all this type of information and annotations into the experiment in an automated way. So to conclude, we have developed a computational method that makes use of reference CytSeq atlases to automate and to improve the phenotypic resolution, accuracy, and reproducibility of immunophenotypic studies. Our software is available in the GitHub web page of our lab. So I will just thank my lab, and in particular Emma Tracy, who did all the experimental work of this project, and Kaya Gobek, who did all the computational work in the project. And she's having a poster in this conference, so I encourage you to visit her poster if you are interested in hearing more details about Stevia. Thank you very much for listening, and I'm happy to take questions.